I want to thank the organizers for having me. I've enjoyed this a lot. For those of you who are about to experience postprandial sleepiness, I've talked about statistics before. I won't be offended. These are my disclosures. Everybody needs a statistician, so I have a lot. I want to start with saying more observational data are needed to plan advanced trials. And we have some growing data. We've heard about a lot of data here. But in the grand scheme of things, in the progressive realm, the data is limited. And we want to know what are the major areas in which the disease impacts individuals at various stages. We've just talked about that in a very general way. We had a very specific topic of hand function, and we know that there are other things that may be important. What are areas of change that change most often and most rapid? And finally, what are the durations that are needed for these changes? Because that will drive what we do in clinical trials. So a little bit of literature, some data from the natural history study of MS from Ontario. Median times to DSS 6, 8, and 10. 12.7 years, 20.6, and 43.9 years. Time to those, that means the time between 6 and 8 was almost 8 years. We talk about doing studies for 2 years. Time to sustained ED, this is from the British Columbia group. 6 was from sustained EDSs, from the onset of SPMS was 8 years. To 8 was 16 years. Again, the difference between 6 and 8, about 8 years. So different populations, same result. This has major implications for using the EDSS as an endpoint in any trial. What about how long does it take to go from 6 to 7? It looks like about 8 years. This is from the Italian group. And what happens with age? These are 54 and 63. Now that is the new middle age. For some of you, you don't understand that. For some of us, it's true. Comorbidities increase with age. And when we're measuring symptoms and or disability, as was pointed out a minute ago, they may be due to other causes. There are a lot of other causes, and they may be causes that are not responsive to putative MS treatments. So you have to think about, what if I have deterioration that's not due to something that's going to be moved by the treatment that I'm using? And that will be in both groups in a randomized trial, and it'll water down the differences that you're going to see, which means you may think you can do it with an effect size that's this big, but because of these other characteristics, it's going to be cut down, and the question is how much? And the services that other people have to go to increase the burden. So patients might be seeing somebody for their arthritis or their cardiovascular disease, and that's going to impact them in many ways, from compliance to attendance. They may drop out of your study because they just can't handle all the visits. And it affects the design of the trial when you're thinking about how great you're going to be. 2 posters I want to talk about that were presented at ECTRIMS. Both were on the same instrument, which I'll show in a minute. My conflict of interest is I was on both of these, and one of them was maybe both of them, I think, were funded by Roche in some way. The purpose of this was to look at early onset, the Ensemble Study, which is an ongoing trial by Roche. I don't own any stock. It's not that I'm selling Roche. It's just that they took patients who had a clinically reported relapse or MRI activity in last month, but they were basically newly diagnosed, and the casting, which they had to fail 2 drugs to get into this trial, but they're still relapsing remitting. So you have early onset and late onset. And the instrument I'm going to show was able to show differences between these 2 groups that you could actually see in this questionnaire. And the questionnaire is called a symptom screen. It does combine a little bit of activities of daily living and how various functions impact you. And it's a 7-point Likert scale, which we have all the time, but the difference is the wording up here. It might have been better to use what Bernard was talking about, which is how much does this affect you rather than how much does it affect what you do. But this is sort of interesting because it does get at the patient-reported perspective. It takes about 2-3 minutes to do. And it actually separates populations. This is from the combined populations, about 600 people from NYU and St. Barnabas. This is Ilya Kister's population. The mean score, you just add those scores up on that questionnaire. The mean score is about 8 for the CIS and RIS patients, about 15.9 for RRMS, and these are RMS of varying durations. You can see down here the durations, 1 year, 8 years, 9 years, 19 years. But there is a dose response. That is, the older you are and the longer your disease duration, you have more and more of these symptoms. And they have problems that affect you, that bother the patient, that I would argue probably are worthy of being an endpoint in a trial if you want to talk about clinical meaningless. A clinically meaningful change on your MRI is good for you to explain to your patients, but it may not have any meaning to the patient. It's by transfer of your knowledge that it has meaning. These are things that actually have meaning. What do the scores look like for those same groups? You can see the red and yellow are the primary progressive and secondary progressive. And just as you know clinically, this is the overall. I've just divided it by 10 so I could get these on the same scale. But what you see is an excess in both of the progressive forms, but the red has the most. So the secondary progressive patients are experiencing more, probably due to the longer duration of disease. But they both look very different. And what are the major ones? The red arrows are pointing out walking. Well, we know that. We just heard that between 4 and 7, it's a walking scale. So they both have that in excess. But we also see hand function, spasticity, and fatigue, and cognition. These are self-reports, but they're probably meaningful. If you look at the ratio, if you wanted to know whether or not what's important relative to relapsing remitting, I was looking at things that were around 1.5 to 2. The yellows are about 2 times, that the report is twice as high in the progressive forms compared to relapsing remitting in walking, hand, and bladder. Spasticity and fatigue are up there. They're the next highest. And I'm looking for consistency between the two. But what you see is that we don't know how to measure bladder function very well for a clinical trial. It's long been known that it's important, but we don't know how to measure it. It might be a very useful measure if we could figure out how to do it easily and simply. This is doing it with a questionnaire, which is pretty easy and simple. This is the change that occurs over a little more than a year on average. And the red lines show what are the functions that are changing in a 1-year period. Hand, spasticity, bladder, and dizziness. Just reporting data, but again, it's showing that we may be able to use things. Spasticity is a little bit hard to measure. We don't have very good measures of spasticity. And I just said we don't have measures of bladder function. Dizziness is a little bit difficult as well. So hand function looks to be a fairly sensitive measure to change, which is what we often want when we design a clinical trial. Well, for designing trials, if we know what our data look like, by the way, designing trials, some people like to think about that as a statistical problem. It's not. It's not a statistical problem. We have a bag of tricks that we dig into, and we work with you to find out what can we use to answer your question. So what is the question and how you ask it is the critical issue. So an old story is told about a disease that strikes a community, kills many people, but some recover. The victims lapse into death-like comas. and it's hard to really know if a person was dead or alive. So, of course, the IRB, or Research Ethics Committees, came in. They were very concerned over burying people alive. So, there we go. So 2 solutions are proposed. One is coffins should be stocked with food and an air vent installed so that, just in case they're alive, it would be ethical for them. And of course, the other is coffins are equipped with a 12-inch stake mounted at the side of the coffin, exactly at the level of the heart. When the coffin is closed, all certainty would end. So, these solutions are answering 2 different questions. One is, how can we assure that we do not kill people who are alive? And the other is, how can we be sure that everyone we bury is dead? Now, that seems kind of silly, but it's not. We ask questions about, is this treatment as good as that one? Or does this treatment work? Those are 2 different questions that require 2 different designs. So the question matters. It matters a lot. What is the goal of the trial? Is the treatment or a disease modifying or symptom management, or both? Obviously, disease modifying treatments often make symptoms better. They may not. If you can stop disease progression, a person may not get better, they just don't get worse. If you have another type of treatment where you actually do make them better, and the progression stops or the disease doesn't advance, i.e. you cure them, then that is a very different kind of goal. Is the goal selection of treatments or doses for a definitive trial? We do a lot of phase 2 trials, many of which don't go anywhere. And do we want evidence to allow for a treatment to be used in combination with other treatments? MS is a unique disease because we don't do a lot of combination therapy yet. We may get there someday. The risk may be too high. I guess the Pyotin trial is one trial that is looking at an add-on therapy. We're probably going to see more of that as we try to approach different ways of getting to the systems that are affected. This is the one that's been driving a lot of the system. Is the goal to allow approval of a treatment by regulators? And I argue that that hasn't failed us in the sense that there are more MS treatments probably than any other disease in neurology, but it's constrained a lot of development. Obviously, the EDSS has been, the IMPACT trial was sort of where it was because the regulators, the FDA said use the EDSS, and Biogen said, no, we can get it done with half the sample size using the MSFC, and they sort of said, don't disagree with us after the study was done. You might have a combination of these, but what is the impact of those? Suppose, I want to go back to my research ethics committees because I end up talking to them a lot. And the FDA and regulators have a societal perspective, and you as clinicians have individual perspectives, and those are very different. So suppose a new approach for a treatment for advanced disease offers an improvement to arm and hand function. And responders in a pilot study that you did was about 60% responded. Forget how I define that. Historical data that you're able to dig up suggests that only 15% of a placebo response. And if you look in another clinic records, though, you found out that maybe 25% can do it if training is done. Maybe there's a training effect. So it could be as much as 40%. It could be 25%. What are the design options for a phase 2B trial? That is, we're going to see if we can use this treatment. Well, one is you can test against a historical 25% rate. You can test against a putative 40% rate. Be conservative, we'll be really sure that it's good. You can test with a placebo group at a 15% rate, a placebo group at a 25, or maybe even an active comparator at 40%. All of these are different design options to answer your questions, which obviously using a single group, everybody talks about how single group studies have drawbacks. They do, but they're a lot quicker because you can use a smaller sample size. If you use a single group against the 25% rate with 17 patients, and 80%, you can have 80% power. Very small, quick study. If you test against the 40% rate, if that's your hypothesis target that you're going to beat, you need 49 patients. Almost 3 times as many. If you use a placebo-controlled trial at the 15% rate, that is a bigger difference than even this, you still need 36 patients. You need 18 patients per group. You have to have more patients to cover the variability that's going to occur in 2 groups. If you go the 25%, you've got 74. And if you go against a 40% rate, you need 218. So the sample size is an order of magnitude different, depending upon how you formulate the question and what you're going to do. So we talked a little bit about how long it takes to get something, but there's another impact to disease worsening and the slowness of disease worsening over time. If you think you're going to achieve a 50% reduction in progression or change over time, if you do a 1-year study, the rate might be 5% to 2.5%. To detect that kind of a difference, you need 906 patients per group. If you wait until 2 years, this 5% continues to go on, so you get 10%. You get a 50% reduction, that's against 5%. Now you only need 435 participants per group. So as time goes on, the absolute difference is widening. And that cuts your sample size. So the duration of the trial is very important. This is the same argument with a 30% reduction, but you can see in a 1-year study, it's almost 3,000 patients per group, and waiting and doing a 2-year study cuts the sample size down by more than a half. It's down to 1,356. Now, somebody can say, well, I can get this study done more quickly. Well, maybe it's not true. And of course, this is why you'll see time to event studies where you follow some patients for some period of time, some much longer than the others, and you wait until you get the right number of events to do your calculations. But there are drawbacks to that. There are assumptions to that, and it's called the person years assumption. It says 1 patient followed for 10 years is the same as 10 patients followed for 1 year. And that's a strong assumption. It's the best we've got. It's how we do all the life table stuff that we do, but it is an assumption. What other designs could we do? Well, first of all, people like to talk about new designs and the benefits of new designs and the wonders of new designs. Well, there's nothing wrong with the old designs, frankly. Parallel group designs, superiority, non-inferiority. I just talked briefly about time to event designs, but they're very efficient. Repeated measures design, we get some power for measuring people multiple times. We get rid of some of the error in measurement. Adaptive designs, I'm going to talk a little bit more about those because they're buzzwords. Some about futility designs, but we seek new designs because we think this is a faster way to make progress. And the answer to that is maybe and maybe not. Just because it's a new design and everybody's talking about it, it may not be the best way to answer your question. That's why I say statisticians have a bag of tricks. We try to pull them out and we try to get the right design for the question that's being asked, not the most vogue design because everybody thinks that's the way to get funded. Let's talk about adaptive designs. They're very vogue, but they mean a lot of things. So it means basically you're conducting a trial in incremental stages and you access accumulated data with predefined decisions. So adaptive designs, phase 1, phase 2, phase 3 is an adaptive design with gaps in between. You do one, you look at the data, you design the next one. It's still adaptive. You're using the information in the prior design. So they're not new in that sense. What is new, though, is the assumptions. If the assumptions in a design that you build in and you check them along the way and find out they're wrong, you can adapt your sample size based upon those assumptions. That you made an incorrect calculation. The variance I estimated was smaller than I've observed. I now increase my sample size to compensate for that mistake. I haven't looked at the results. I've looked at the assumptions. Allocation rules, we often do that. We try to balance within clinic. Those are adaptive designs to make sure that we have the right number of patients in the right places, maybe with the right characteristics by stratification. In sampling rules, we do that. The number of subjects we use in the next stage may depend on what we've observed here. We may close certain strata. We want so many secondary progressives in the study, but we also want so many primary progressives. In stopping rules, these are where a lot of discussions are held now for stopping for efficacy, for harm, and futility, and often about decision rules. Do we go forward? What is the decision rule that we're making in order to use this adaptive design? Some cautions, just because it's so vogue. The logistical issues are critical to adaptive designs. You have to know what you're going to do with the data. It's not, I'm going to look at it and decide. It's not only because of regulators, it's in general probably the right thing to do to know what your changes are going to be. How are you going to adapt? What are the rules you're going to use? When are you going to increase your sample size? You need to have the money in the bank to do this. So you have to be funded in advance. We're doing a hypertension trial right now with an adaptive design that had $1.8 million in the bank in case we needed to increase our sample size. Obviously not an MS study. You want the adaptation to be formed, to be decided by someone who doesn't have a stock in the study. You want them to be independent of a conflict of interest. And remember now, you're talking about, well, you know, everybody wants their trial to work. Rarely do people go into a trial saying, gosh, this is going to be great. I'm going to spend 5 years of my life and this is going to fail. So they all want it to work. So that bias is to increase the sample size. And there's a lot of those. So you want someone who doesn't have that, and you only want people, if you're going to do what's called an unblinded sample size assessment, you want the minimal number of people because information leaks out. Once it leaks out, it could affect the rest of the study. It's why when we talk about monitoring data that we actually have various statistical procedures to control the type 1 error. It's not because the data are going to change. It's because the people who know about the result may change their behavior, and in that sense, they will change the p-value. This recent paper, access to interim information of the shape of the survival curve. This was looking at an adaptive design for survival type analyses. And they actually found out, given the lack of efficiency advantage over group sequential designs, sample size adjustment designs and time to event studies remain unjustified. It's a pretty strong statement. And there's a number of statisticians who have found in most designs, group sequential designs are equally valuable, and you don't have to go through all this logistic rigmarole. You just plan them well in advance. I think adaptations to correct the sample size for assumptions gone wrong is a good idea. And just to use it to answer the question, how many more do I need to observe a significant p-value, I think can lead us into trouble. I actually don't think the characteristics of these adaptive designs and designs that are moving toward a more significant result based upon the interim results have been studied enough for the impact on type 1 errors. I think we might be overestimating the benefits of the treatment that we end up with after we've done these. A few designs I'm going to talk about at the end very quickly. Futility designs, targeter enrichment designs, two-stage, individual endpoints, goal attainment, and choice designs. And I'll just move through these quickly. First of all, a futility design and a futility trial are similar to non-inferiorities, but they're not the same. A futility analysis is not the same as a futility design. What you do is you assume a negative outcome in a futility design. You assume that, as you would in a non-inferiority study, that it's worse. And then you try to reject the hypothesis that it's worse and come up with the idea, this might be worth studying, it's not worse. You look at drug A is worse than drug B, and what we do is we often use historical controls or a single group for a single group study because we want to get this done quickly. Otherwise, there's little advantage for it. And we must reject futility in order to move forward. What are the reasons why the pros are opportunity costs. Spending money for a full-fledged futility trial and carrying it all the way out to the end when it could have been seen early that it was useless will save money and time. And time is probably the most valuable asset we have. And it's ethically wrong to continue to recruit patients to a study where there's little hope of achieving the endpoints. The cons are, well-conducted trials provide valuable scientific evidence even if they come up negative. And if you do the entire trial, you might really learn something. The cost of designing futility trials with the planned futility analysis may outweigh the savings in time and certainly in money. And will it really save money? Because when you get done with the futility trial, you're still doing the larger scale study to test whether or not what you have. And depends on how long this futility trial takes to run. Target designs are things that you go on a biomarker. Now, those have been used in MS fairly successfully, but maybe you select a target population, like someone who's had relapses in the last 2 years, or they have a GAD lesion on their MRI to enrich the population. And the purpose is, to look at the treatment for people with that marker. So everybody in the population has the marker. And the intent is not to validate the marker, but it's rather to enrich the population for responders. Now, a whole class of these are done in cancer called umbrella and basket designs. And they are basically, I have a single drug that will apply to all kinds of cancers, or a single drug that will apply to all kinds, I take all comers in MS. And the other is, I take all of this type of patient and I prescribe a whole bunch of treatments and then get rid of ones that don't work. One's a basket, one's an umbrella. They're very useful, but the efficiency of these designs is based on the portion of patients that have the positive biomarker. And the effectiveness of the treatment compared to the control for the biomarker negative patients. So when we think about MS trials that might have enriched on the basis of someone having a GAD lesion at baseline, well, it will require fewer patients when less than half the patients are biomarker positive. And the treatment has little benefit in those negatives. So those people who didn't have a GAD lesion, the 60% that you find that didn't have a GAD lesion, those are the ones that shouldn't get any benefit from the treatment. And in fact, that doesn't work in MS. And therefore, it's not really valuable to do that kind of biomarker trial in MS. Sliding dichotomized what are called individualized endpoints in stroke, for example. Patients with larger volumes of damaged tissues have poor prognosis. So what you might hope for them is that you can stop them from having anything other than moderate disability. Whereas patients with smaller volumes on their MRIs, you might say a desirable outcome is to have complete functional recovery. This idea might be very useful in progressive disease, where the effects of individuals varies greatly, and you set up a target for each individual to have some outcome. And it can get more complicated, which what are called Ghoul attainment scaling studies. And here, what you have is each patient basically has something like an MSFC. It's a combination of the things that are bothering this patient. Each person has one, and they're all mapped in a pre-specified way. It's not getting the data and then figuring out how best to combine them, but you specify in advance. So it might be the improved use of hands for one patient might score a 1 or 2 on a 7-point scale, and walking with a cane from a walker might be mapped as a 1 to a 2 for another patient. And then you have all of these ordinal outcomes. Each one is individually tailored to the set of disabilities that have affected a patient. It allows this patient-centered outcome to be used, and it enables larger sample sizes, not by design, but it enables a broader patient population to be included so that you're not restricting to patients who only have this much disability in this left arm function. So it gives you a broader way to approach a population, which makes more people eligible for doing clinical trials. 2 or 3 stage designs. These are where you actually build in, they're an adaptive design, but they build in a futility endpoint. If I don't get so many, it's very good for testing new drugs in small populations. You set up a hypothesis, it must achieve this level of success or I won't continue. So you might be able to do it in N1 patients, and if you don't get that many to respond, you stop. So you need a response marker that's relatively quick. So something like change in hand function, et cetera. And it doesn't allow you to stop for efficacy, but it does stop you from following treatments that are not going to go anywhere. This is called a choice design. And it's often difficult to recruit patients to this. Many people obviously want something. And placebos are often very difficult for people to do. This is a design that Rob is doing in exercise, where the patients get randomized to a choice arm and a randomized arm. The randomized arm, they get home-based telehealth for their exercise intervention, or they get a facility-based exercise intervention. And the other part of this choice arm, if they get randomized to the choice, they get to choose. Do they want to do it at home or do they want to come to a facility? And these are kind of interesting designs. You get a treatment effect, the average of effect of a particular treatment in a specified population. So it's sort of what would happen in the real world. So it gives you what Gavin's been worried about called estimands. It gives you the estimate in the total population where people get to choose what they want, and some people might get prescribed what they want. It gives you this selection effect. What is the difference in the treatment effect that's influenced by self-selection of a given treatment by patients? And what is the preference effect, the change in the outcome resulting from the interaction between the patient's preferred treatment and the treatment they actually received? We're doing one of these in a bone study in neurofibromatosis because there are some people who know about this drug, BMP, it's actually a device, and they want it, and they refuse to be randomized. So we allow some people to choose, and we'll see what happens, and we have some places where it's not available, and therefore those will be randomized, and we can try to make inferences from these two. So it's a new class of designs that in many circumstances actually has a lot of merit to encourage more patients to participate in studies. So finally, the increasing complexity of later stage disease requires more planning and actions during the analysis phase. And what that means is when you think about doing your study, give us statisticians time to do the analysis correctly. Don't call us the night before the abstracts do. Really try and really spend time realizing that analysis is an iterative process. It involves you, it involves a statistician. Yes, it involves a computer, but it really is a cognitive. Thanks.